the next topic, which is Winter on Forage Barley. Dr. Jamie Sherman will provide us an update on new releases and plans for the future. Dr. Sherman is an assistant professor at MSU, and she is our barley breeder. And she's been a great collaborator with the Research Center as well, and around maize as a whole. And so welcome, Jamie, and thank you for this presentation today.
Great, thanks for the introduction. Can everyone hear me and see my slides?
Yes.
Okay, I'll go forward.
Yes, good.
Good, thank you. So recently, our collaborations with the your center has focused on forage barley development. So that's going to be the focus of my talk today. Before I get into that, I would like to thank our sponsors, which I just went through very quickly. The Wheat and Barley Committee is important, a supporter of our research. And I'll mention a few other sponsors as we go on. And also our cooperators, so all the research stations are important in running our trials. And then we have some specific collaborations with some of the research center and faculty at MSU Bozeman. And so we appreciate that. It makes the program so much stronger. If we didn't have those collaborations, we would not be at all successful. And then this is our team. So it's a large team, and we aren't even representing everyone here. The staff members are myself, Hannah Turner, who runs the quality lab, and she has an assistant, Sarah Olivo, and then Greg Lutkin is our new field technician. And then the rest of the team is some very important graduate students. I'll talk about one of their work today, and then undergraduates as well. So the workhorses really are the undergraduates who help us process everything and gather all the data. You can see here are the breeding goals that we established since the inception when I took on the barley breeding position. And you can see that from the beginning with the spring program, we have been working on forage barley development. We also work on malt development, feed, and food. And so, and you can see that there's a variation in what we're focusing on at all those different levels. But all along, we've been working on developing a new forage line. And recently, we instituted winter breeding for barley in hopes that we could develop a winter barley suitable for Montana. And one of our first goals is to release a winter forage barley. But we're also working on malt. And of course, the primary focus there is just to get something to survive the winters in most of the state. So we do have a upcoming release. And in this slide, you can see while I'm comparing this experimental line that ends in the numbers 2902 with varieties for several different traits. And that's how we decide whether a line should be released or not. Is it better than what's currently out there for the growers and producers? 2902 is from a cross between Lavina and a variety called Cowboy that was actually released in Wyoming. And so the first column of numbers there is the amount that the variety scores, for example, for grain yield or forage yield, protein, etc. And then the second column is how the new line scores. And then we do what is the percentage of that new line to the variety that we're comparing it to. And so what we're seeing there is that this new line is statistically equal in grain yield to current lines Levina and Hayes, and then is an improvement on the new line. It yields, grain yields a little bit better than Haymaker. And you might wonder, well, why am I worried about grain yield when we're going to cut this before the producers are going to cut or graze this before it goes to grain? Well, we need to have good enough yield so that the seed producers can produce it economically than to give the seed to the growers. So it needs to be at least as good, and it is in grain yield. And then where we see the big advantage, probably mostly due to its greater height than Levina and Hayes and Haymaker, it is an improvement in forage yield, a significant improvement. And then it's equal to in the percent protein and digestibility measures of NDF and ADF. So it should be nutritionally equivalent, but higher forage yielding than existing lines. And so we hope we can release this variety relatively soon. We would like to have another year's data comparing it to Haymaker. And then we also need to produce breeder seed so we can give it to the foundation. And so that will take another year before we can release it. So then another thing that we're working on, and thanks to Clint for growing this for us this year, we are trying to develop, as I said, winter forages. And so why would we think that a winter forage would be helpful? Well, they will increase, they could have more biomass. They could help the growers by allowing for an earlier harvest. Our winter lines that we're working on right now can be harvested 2 to 3 weeks earlier than winter wheat. They could provide winter ground cover for the year. They have access to early spring moisture, so that could be an advantage, and they can avoid some late high temperatures, which can increase nitrate production. They could allow for spring grazing. So if you don't want to cut them, potentially they could be grazed early and then maybe would recover later. We need to test that, but that's kind of an interesting possibility. And this year, NWARC is growing 52 early generation bulks. So we're in the early stages of the winter barley program. We've made crosses and we've inbred them. And now we're trying to start testing them. And what we're going to do is put them out as an earlier generation and then select the lines that survive from those 52 bulks. So it's a little bit different process than we use with our spring program, but we're trying to jumpstart this program. Now, those 52 separate bulks come from actually 4 crosses. And 2 of these crosses involve lines that come from the Vavilov collection. And I've talked to you about Vavilov before. He was a Russian scientist who traveled the world collecting plants that he thought would be important for the future of agriculture. And in part of that work, he collected cold tolerant material, including winter barley material that was cold tolerant. And so it's our hope that by introducing this germplasm, incorporating it into our breeding program, that it will provide more cold tolerance to our current material. And of course, we know the story of Vavilov, that he got crosswise with Stalin and was jailed and died of starvation in jail during World War II. And his compatriots, his workers in his institute, at risk to their own lives. In fact, many of them starved to death, protected the seed. So to me, it's a sacred thing that we have this seed, that people died to get it to us, and we hope that it will provide an advantage to the growers of Montana. And I think it's good for us to know that history, that people thought this was so important and essentially sacrificed themselves for it. So that barley work is in the process. It is at early stages, but if you go to the farm this summer, you should be able to see these bulks out there where we will be able to select lines to move forward. And why we need this really is, to ensure that this material can survive across the state. We really need to have all these different locations to test it. So we really appreciate Clint's willingness to help us with that. Okay, so then I just wanted to briefly talk about some of the research that we're doing that we hope will help improve our chances of creating a good barley forage. This is a graduate student project, and one of the problems with forage work is that it's extremely labor intensive. Collecting all of the data points, including, the forage yield, you have to take grain yield, you have to get material that you're going to run digestibility studies for. And so what we're hoping in this graduate student's work is to use drone imaging to collect that data, so that it would be easier for our, the stations to grow this, and it won't be such a hard process for them to have to go through. So we're excited about the possibilities here. We're hoping that this will work. And kind of our next step is to empower the stations to get this technology if they don't already have it. So we're really excited about the potential here. One of our stumbling blocks is it's easy to gather the data. It only takes about 30 minutes to fly the field, and then we've got all these data points. But then taking it from that to usable data, that's been a stumbling block. And New Belgium actually helped us buy the drone we're currently using. And now they're helping us get the software that we think we need to make this a more high throughput process of converting the data from these images to numbers that we can actually use. So we're hopeful that once we work through that, then it'll be ready for the stations to take it on and save time and energy in gathering that data. Also from Tracy's work, we have found that we have proxies for forage traits. On some of these, you think, it makes common sense. For example, we get more tons per acre. Oftentimes when we have taller plants, which is the first square there, but we also get more tons per acre if we have later heading. So that kind of makes sense too. The longer we stay in the vegetative state, we can grow more vegetative material. So delaying heading can give us more tons per acre. It also, however, might reduce yield. So that's a trade-off. And then also delaying maturity. So going from heading to the grain developing and that taking longer also helps increase tons per acre. So then we were wondering, well, what's the relationship with these traits with how digestible the material is? The digestibility is really dependent on the fibrous material in the cell walls. The more fibrous material like hemicellulose, cellulose, and then lignin, the more of those materials, the less digestible it is to the animal. And so it kind of makes sense that and ADF is measuring the actual amount of fiber left. So we want ADF to be low. We want there to be less fiber, so it'll be more digestible. And so it makes sense that as the plant gets taller, it needs more fiber to support itself. So there's that relationship. But we also see a relationship between heading and other maturity traits where we get more digestibility the later heading is. And this is exciting because this means we can select for later heading, we can increase tons per acre and digestibility as well. And the student is even looking at this at the genetic level to identify genes that impact these traits. And sometimes for some genes, we see this pattern where we see that as we increase forage yield, we reduce digestibility, we get more fiber, so that's not good. But we have identified some genes where we can get more yield. So over on the A, under forage yield, the AA gene gives us more yield, and then it also gives us more digestibility or less fiber. So that's the sorts of genes that we want to go for in this trying to improve forage, because we can have our cake and eat it too. So that's exciting. Okay, so that gives you an overview of some of the things that we've been working on. And I guess I'd like just to have a conversation now of so that you can ask questions and we can talk, so I can learn from your perspective about what we should be working on for forage barley.
I would have a comment real quick, Jamie, or just something to maybe be thinking about, is it always seems like we're struggling for quality versus yield. Doesn't really matter when we're in any kind of hay oats, hay barley kind of thing. We're always like, well, I want maximum yield, but I want maximum quality. So I guess my thought would be is, maybe working on things that, we can still cut the yield without jeopardizing quality might be something to look at.
Yeah, I'm hoping some of those genes that I was showing you will allow us to improve both traits. I'm also submitting a grant to USDA where I'm wondering if, doing a mixture of crops, like a winter barley and winter pea, or I don't know, would allow us to have both as well. So, that's something else that we're interested in starting to think about. Like, doesn't have to be a monoculture for forage. Maybe we would be better off with some mixtures, which I'm sure some people already use, but we haven't really bred for those mixtures. And that's one of the things that we would like to correct is to start breeding for some things like that, that would be some specific mixtures useful to growers. It could also very well, if we did mix a legume with a cereal, then that could help with decreasing nitrogen inputs as well. So there's some big advantages of that. So we're trying to think about breeding for systems. System approach breeding is really what I'd like to do. So, eventually we would like to talk to the growers, what systems are you currently using? What should we be breeding for if we're looking at it as more of a system approach? Are there some species that you would be interested in intercropping, or would you be interested in grazing directly? And those sorts of questions would help guide us for what we should be breeding for.
I think keeping nitrates under control is another issue that would be interesting to look at.
Yeah, and we do have the ability to measure nitrates in my lab, and we do that. One of the issues in that is controlled a lot by the environment. So how much genetic control we have over that. So, it may be more of a management issue, like Jessica was talking about. So I think that is something that we need to constantly keep in mind, that we need to consider.